Welcome to our webinar on Mosaicism and Chimerism. This webinar is presented to you by Priyal and Sophia, students in the Medical Genomics program at the University of Toronto. In this webinar, we will explore what Mosaicism and Chimerism is, how they occur, how they differ, how they are detected, and how they impact health. You may have learned that every cell in our body carries DNA. But have you ever questioned if every cell in a person truly carries the same genetic code? In some individuals, different groups of cells can carry different genetic instructions, either arising from early developmental changes or from the convergence of cells originating from more than one individual. Even though we often picture our DNA as consistent across every cell, Mosaicism and Chimerism remind us that biology is full of exceptions. In the first few minutes, you will explore Mosaicism with me, Priyal.
Then you'll explore Chimerism with me, Sophia.
So what exactly is Mosaicism? Mosaicism means that a single individual has two or more genetically different cell populations, all originating from the same fertilized egg. In Mosaicism, some cells carry the usual DNA sequence spelling, while others carry a genetic variation, a misspelled version of the original DNA sequence. Imagine a tiled floor. Most tiles follow one pattern, but scattered among them are tiles with slightly different designs. That's Mosaicism. Now, how does Mosaicism occur? Mosaicism arises when a genetic variant is introduced into DNA being copied for cell division. Every cell that develops from the cell carrying the variant will inherit the change. Because this change is only present in some cells, the body ends up with a mixture of different cell populations, some carrying the variant and others not. Given that the human body undergoes trillions of cell divisions over a lifetime, it's nearly guaranteed that each of us is a mosaic. So when can Mosaicism occur? Mosaicism can arise at almost any stage after fertilization, from early embryonic development to late life. The earlier the variant occurs, the more tissues inherit it. If a variant occurs during the first few cell divisions after fertilization, large portions of the body may carry it. If it occurs later, say during organ development, it may be confined to a single tissue, like only the skin or only blood cells. Let's explore this concept more by diving into the four major categories of Mosaicism: somatic Mosaicism, germline Mosaicism, gonosomal Mosaicism, and confined placental Mosaicism. Somatic Mosaicism refers to different populations of cell in the somatic tissues of the body. When a variant arises in a somatic cell, this generates a population of cells that are genetically distinct from other somatic cells in the individual. An example would be a variant detected only in blood or skin cells. These variants are not passed on to children because only somatic cells have the variant. Germline Mosaicism is when the variant exists only in an individual's egg or sperm cells. Since the variant is found in germline cells, these variants can be passed to children. In this case, the child would not be a mosaic. The variant would be present in all of their cells. The third major category is gonosomal Mosaicism, which occurs when the variant is present in both somatic and germline cells. This type of Mosaicism often arises very early in embryonic development, before germline and somatic cell lineages have been determined. It is important to mention that there is a less common fourth type of Mosaicism that occurs in approximately 2% of viable pregnancies. This is known as confined placental Mosaicism. It is a tissue-specific Mosaicism that occurs when the variant is confined to only the cells in the placenta, with the fetus being non-mosaic and comprised of the original cell population. In these cases, the placenta may be comprised of both variant and original cell populations, or entirely of the variant cell population. After hearing about these various types of Mosaicism, you may be wondering how or if Mosaicism can cause disease. The answer is, it depends. The consequences of Mosaicism vary widely. Variants that are disease-causing can contribute to disease in a mosaic form, depending on which cells or tissues contain the variant and what proportion of cells carry the variant. We know that Mosaicism has been identified in a wide range of genetic disorders such as chromosomal abnormalities leading to Turner syndrome, Klinefelter syndrome, Down syndrome, neurological conditions leading to some form of Rett syndrome, and skin pigmentation disorders such as hypomelanosis of Ito. In these diseases, being mosaic for the disease variant can result in a milder presentation of the disease because not all cells are affected. In fact, some diseases only occur in the mosaic form because they would otherwise be lethal. Another important category of mosaicism-related diseases is cancer. Most tumors develop from variants that arise in somatic cells of the body during life, making cancer one of the most common examples of somatic mosaicism. Finally, mosaicism can also affect an individual's likelihood of having a child with a genetic condition. For example, often individuals with germline mosaicism are unaffected by a disease-causing variant confined to the germline cells because the somatic cells of their body, the ones that make the body function, do not contain the variant. Although, since the variant is found in germline cells, these variants can be passed to children and cause disease. In this case, the child would not be a mosaic. The variant would be present in all of their cells, causing disease. Germline mosaicism will not be detected in a parent undergoing a DNA blood test to determine reproductive risk because blood is somatic tissue. Consequently, they may not realize they are a carrier for a genetic disease. Understanding mosaicism is becoming increasingly important as sequencing tools improve our ability to detect these subtle genetic differences across tissues. Since mosaicism can be limited to specific tissues, a variant may be present in one tissue but absent in another, making detection a challenge. For example, a disease-causing variant may be in brain cells but not in blood cells, so a blood DNA test might miss it. Additionally, if the variant is only present in a very small proportion of sequenced cells, it may be mistakenly overlooked as a technical error in sequencing. Clinics and laboratories commonly use methods such as karyotyping, fluorescent in situ hybridization, and chromosomal microarray analysis to detect mosaicism. The most sensitive method to detect mosaic single nucleotide variants, insertions, deletions, and low-level mosaicism is next-generation sequencing. In next-generation sequencing, mosaicism is identified when only a small proportion, about less than 25%, of the DNA sample contains the variant. Before we talk about chimerism, let's look back at what we've learned today about mosaicism. Mosaicism is the presence of genetically distinct cell populations within one individual originating from the same cell line. It occurs when variants arise during cell division. It can happen any time after fertilization, with variants occurring earlier in development affecting more tissues. We discussed 4 types of mosaicism: somatic, germline, gonosomal mosaicism, and confined placental mosaicism. Mosaicism can lead to a range of genetic diseases, sometimes explaining milder severity of diseases, and it is often detected through DNA sequencing or chromosomal microarray analysis. Let's take a few minutes now to test your knowledge about what you've learned about mosaicism. Feel free to pause the video if you need more time to answer. A variant occurs at the 8-cell stage during embryonic development. Which outcome is most likely? A. The variant will be confined to one organ due to complete lineage restriction at this stage. B. Only germline cells will contain the variant, not somatic cells. C. Only somatic cells will contain the variant, not germline cells. Or D. Multiple tissues will show a variable level of the variant. The correct answer is D. Multiple tissues will show a variable level of the variant. Let's try another one. Why is detecting mosaicism challenging? A. You need to test multiple family members. B. The tissue being tested must contain the variant. C. We don't have a good understanding of what mosaicism is. Or D. You can only detect mosaicism at certain stages of development. The correct answer is B. The tissue being tested must contain the variant. Let's try one last one. A woman undergoes genetic testing for a known familial variant causing a dominant cardiac disorder. Surprisingly, the variant is detected at a low frequency in her blood sample, but is absent in her saliva sample. She has no symptoms and both her parents test negative. What is the most likely explanation for these findings? A. The patient inherited the variant from a parent who is an asymptomatic carrier. B. The laboratory result is likely contaminated because the allele frequency is low. C. The patient has somatic mosaicism affecting only certain tissue lineages. Or D. The patient has germline mosaicism affecting only the gamete cells. The correct answer is C. The patient has somatic mosaicism affecting only certain tissue lineages.
Now that we're experts on mosaicism, let's move on to the next topic, chimerism. Chimerism is a genetic phenomenon where a single individual is made up of cells from at least two genetically distinct zygotes. This means an individual is made up of cell populations with different genetic origins.
This differs from mosaicism, where an individual is made up of cell populations originating from the same genetic origin.
You might be wondering, what causes chimerism? Well, there are several types of chimerism, and they can roughly be divided into three categories: microchimerism, natural chimerism, and iatrogenic chimerism. Let's begin by diving deeper into microchimerism. Microchimerism is defined as chimerism where the minor cell population makes up less than 1% of all cells in an individual. This means a second cell population is present, but only in a very tiny amount. Microchimerism can happen in a few different ways, but by far the most common cause is pregnancy. During pregnancy, the placenta acts like a bridge between the mother and the developing baby. It's responsible for exchanging nutrients, oxygen, and waste. It also allows cells to cross between mother and fetus, with more fetal cells being transferred to the mother. When fetal cells remain in the mother's bloodstream and tissues, this is called maternal microchimerism, and is quite common. On the other hand, fetal microchimerism refers to the presence of maternal cells in the fetus, but this is much less common. On the other hand, macrochimerism occurs when the minor cell population makes up greater than 1% of all cells in an individual. However, it is simply referred to as chimerism. This type of chimerism can broadly be divided into natural causes and iatrogenic causes. Let's start by learning about natural chimerism. The first type of natural chimerism we are going to discuss is dizygotic fusion chimerism. It is also referred to as dispermic or tetragametic chimerism. Dizygotic fusion chimerism occurs when two separate fertilized eggs, called zygotes, fuse in the womb. This fused population of cells develops together to form a single individual. This means that one individual is made up of cells from each of the original zygotes. The other natural cause of chimerism is called dizygotic blood chimerism, also known as twin chimerism. Normally, dizygotic twins, commonly referred to as fraternal twins, each develop from a separate placenta. In rare instances, their placentas can become fused during pregnancy. This connected blood system allows blood-forming stem cells to pass between the twins. If the cells persist in the other twin, they can create a stable population of blood cells. This results in twins containing a mixture of their own and their siblings' genetically distinct blood cells. This type of chimerism is one of the easiest to detect from genetic testing because a dual genome is present in the blood. Note: if less than 1% of the total cells are comprised of the minor cell population, this form of chimerism is considered microchimerism. The last major category of chimerism is iatrogenic chimerism, also referred to as artificial chimerism. This type of chimerism is the result of human therapeutic interventions. For example, an individual who underwent an organ transplant is an iatrogenic chimera. They have two genetically distinct populations of cells in their body, their own cells and the cells that make up the organ of their donor. People who receive a bone marrow transplant are considered chimeras too. A bone marrow transplant replaces the recipient's original blood-forming cells with those from a donor. Over time, the donor cells take over the production of blood cells in the recipient's body. So, genetically speaking, their blood is no longer their own. It carries the donor's DNA. Now that we've explored the different ways chimerism can occur, you might be wondering, what is the impact of chimerism on health? Well, with dizygotic fusion chimerism, one cell lineage often dominates specific tissues or organs. Sometimes, but rarely, this results in visible signs like two different skin tones, different eye colors called heterochromia, or combined male and female genitalia. What's interesting is that most people with chimerism don't show any of these obvious signs, so they usually have no idea that they carry a second set of genetic material. Because of this, most cases of natural chimerism go completely undiagnosed throughout a person's life. If chimerism doesn't usually affect a person's health, why does diagnosing chimerism matter? Well, if someone with chimerism isn't diagnosed, an unexpected outcome may occur when they have children. From a genetics perspective, a chimeric mother might be more distantly related to her baby than expected, as if she was an aunt. That's because the DNA passed onto the child could come from the second minor cell population, originally the zygote of her twin. This could result in a false negative parental DNA test if the DNA sample of the parent being tested only contains DNA from the major cell population. With iatrogenic chimerism, the impact on health is quite different, mainly because an individual is made chimeric intentionally as part of a life-saving measure. This means that iatrogenic chimerism often has a positive impact on health, but it can cause some health complications, such as rejection risk. Another example would be genetic testing of individuals with a bone marrow transplant. The DNA sample for genetic testing usually comes from blood. In recipients of a bone marrow transplant, their blood is entirely, or almost entirely, made up of blood matching the donor's DNA. It is important that healthcare professionals are aware of this during genetic testing because a blood sample will not accurately reflect the genetic profile of the patient. But how do we actually detect chimerism? And just as important, how do we tell it apart from something that appears similar like mosaicism? One of the main tests used to identify chimerism is called short tandem repeat, or STR testing. STR testing is a way to look at patterns in our DNA. Our DNA has regions where a short sequence of bases repeats over and over. These repeats are unique to each person, kind of like a fingerprint. If someone is a chimera, they'll have two sets of patterns, or two fingerprints, instead of just one, on all chromosomes in the two different cell populations.
This differs from mosaicism, where the two fingerprints usually only affect one or two specific chromosomes.
Let's take a moment to look back at what we've learned about chimerism. Chimerism is a genetic condition where an individual has two or more cell populations with different genetic origins. We explored the main types: microchimerism, which includes fetal and maternal microchimerism; natural chimerism, like dizygotic fusion and dizygotic blood chimerism; and iatrogenic chimerism, which happens through medical interventions such as organ or bone marrow transplants. While chimerism usually doesn't cause major health problems, it can lead to unexpected outcomes if it goes undiagnosed. To tell chimerism apart from mosaicism, scientists use STR testing. Now it's time to test your knowledge of chimerism. Feel free to pause the video if you need more time to answer. Let's begin. Microchimerism most commonly occurs due to A. Cancer, B. Viral infection, C. Pregnancy, or D. Organ transplantation. The correct answer is C. Pregnancy. Let's try another one. What is iatrogenic chimerism? A. A condition where two embryos fuse. B. The presence of genetically distinct cell populations due to medical intervention. C. The exchange of blood cells between twins during pregnancy. Or D. Two distinct populations of cells caused by mutations. The correct answer is B. The presence of genetically distinct cell populations due to medical intervention. Let's try one more. Which type of chimerism is most likely to be detected by genetic testing? A. Maternal microchimerism, B. Dizygotic fusion chimerism, C. Dizygotic blood chimerism, or D. Iatrogenic chimerism caused by an organ transplant. The answer is C. Dizygotic blood chimerism. This is because dizygotic blood chimeras have two genomes in their blood. DNA collected for genetic testing usually comes from blood, and therefore a dual genome would be detected during genetic testing. To conclude, let's take a look at mosaicism and chimerism side by side.
Mosaicism is the diverging of two or more genetically distinct cell lines from a single zygote.
Chimerism is the convergence of two or more genetically distinct cell lines from more than one zygote.
In mosaicism, only a few positions in the genome differ between mosaic cell populations.
Whereas in chimerism, the entire genome differs between cell populations.
Let's end off on one final knowledge check. Anna gives birth to a child with a rare genetic disease. The mode of inheritance of this genetic disease is known to be autosomal dominant. In other words, only one copy of the variant needs to be passed on to the child from either parent for the child to inherit this disease. Anna and Tom both test negative for this variant, so physicians assume this is a de novo variant, meaning it was developed in the child. Anna gives birth again, and the child has the same genetic disease. What could possibly explain this? A. Anna or Tom has somatic mosaicism. B. Anna or Tom has germline mosaicism. C. Anna or Tom is a dizygotic fusion chimera. Or D. Anna or Tom is a dizygotic blood chimera. The answer is B and C. Anna or Tom could be germline mosaic for the disease variant, or they could be a dizygotic fusion chimera. Since both their children have the genetic disease, it is likely that the disease variant was inherited from one of the parents' germline cells. Germline mosaicism and dizygotic fusion chimerism are the two options where the disease variant could be present in the germline.
Thank you for listening to our webinar on mosaicism and chimerism.